leave it to the nerds, Jack. Okay, so lecture 34. Okay, genetic variation. Okay, these first two figures, it's just so you're clear on the concept, the distinction between germline somatic cells, right? Germline mutations passed on, somatic cells, cancer. Okay, then we get into these mechanisms. How do we alter genomes? How do we shape genomes? How are genomes formed? And there are these 5 different types of mechanisms that we go into. And so we didn't really go much into this mutation within a gene. That's pretty obvious. So then we talked about duplication, and then hand in hand with that was deletion. Then we're going to see exon shuffling, and then this notion of horizontal transfer. So a bacillus genome, that's just to show you that there's a lot of this duplication going on. And then what's the mechanism? Unequal crossing over. That's how you get duplication. You end up also with a deletion. And now we're talking about whole genes. Later we're going to talk about exons. But if you duplicate a whole gene, then you can get new function. In this case, you can go from a monomeric to a tetrameric protein, which allows for the cooperative effect of oxygen and CO2 binding. Okay, so you can't really make a tandem. You can't like fuse 2 genes together and expect to get a new function. So this is just, you make another type of gene. Dulce and I were talking about this. So you go from an alpha to a beta, and now you can form this complex. And then this didn't happen just once in the evolution of our genome, but so you had alpha, beta, and then the beta you had these successive tandem duplications to give you all these different forms. Then Carl the frog story, Carl's tetraploid. This probably happened because you had duplication of the genome, but then a spindle problem, and it remained diploid. And then I guess you had to have a female and a male that are both diploid to get the tetraploid. Okay, so now we're going to talk about exons before we're talking about genes. Exons, same deal. You can get unequal crossing over, and now you can get duplications of exons, or you can lose them. So that's duplication. There's also shuffling that you can see what you can get. It's just all this mix and match of these small domains, and then the mechanism responsible for that is transposable elements. And so this wouldn't be possible without introns, right? So these elements are in the introns. They're going to move the exons. So through duplication and shuffling, then you can get what does it say in the fine print on the shuffling page? So for our 30,000 proteins that are estimated, then that these may have come from just a few thousand exons of very small domains. Okay, so that mechanism of duplication and shuffling is responsible for the structure of most of our proteins. All right, so we saw what transposable elements can do. They can play a role in the shuffling mechanism. They can also, so that's moving an exon. You can also just move then the whole gene to within adjacent to a regulatory region and get these dramatic effects on development, right? And so these transposons then appear to be a significant source of developmental change. So that's a second effect of the transposable element. And then and we're going to see a third one later, or a third role for them. So bacterial cells then, this horizontal transfer, and this is through conjugation. So this is 2 different types of cells, right? Through this bridge, and this is so this is that makes it horizontal transfer. It's not vertical, and it's through this mechanism. It's not just transformation where a plasmid's going in; they're actually conjugating. And then this is responsible for the rapid rise of drug-resistant bacteria in clinical settings. Okay, this promiscuous effect or activity of genetic material in early life. And once that all settled down, there seemed to be 3 branches of life, 3 lineages, and then they had these shared properties with respect to general functions. Okay, life's tree, Artie. So Artie was the missing link between the more modern erect forms of our species, our genus, and the earlier. So Artie. So how do we know about him on a molecular level? Well, we can look at the fossil record and correlate it with the molecular clock. That's that next slide. And then we can even take that as far as getting a very good idea about what the nucleotide sequences of Artie's genes were. And that is by comparing human with chimp and using gorilla as a tiebreaker, right? So you just go through, and if there's a difference between human and chimp, you look at the gorilla, and then the gorilla tells you which one it was. So not only then on the nucleotide level, but just the gross organization of the genome, the chromosomes, all very similar. That's in contrast to the mouse. The mouse, 75 million years apart. Well, Artie was 5. I always have to ask that. I think it was 5. So 75. A lot happened in the meantime. And this was largely due to transposons jumping in. Do they jump into the exons? No. They jump into the introns. So you just look, and wherever it's peppered with introns, that tells, with transposons, that tells you that's an intron. Where it's not, that's a good clue that you're in an exon. And that's because you have to preserve function. So that's like a third kind of effect of transposons, is they're going to lead to kind of resculpting, if you will, of the genome in these introns. Okay, so divergence in the introns. Fugu doesn't have introns. So the human gene has a ton. These transposons have jumped in. It's blown up these introns, so it's a much bigger gene. Okay, then the sequencing, the boring sequencing of the ribosomal RNA turned out to reveal a 3rd branch of life. Pretty big deal. Okay, the human genome, big. Chromosomes have been sequenced. After all of that, turns out only 1.5% was protein coding. Okay, so a few percent of it was, and this was determined by using Sanger sequencing and then computer programs to find these open reading frames. And then if you don't have it in color, this slide doesn't mean anything to you about the preserved order of the genome. So you have to see that with these arrows that there's kind of an orangish, is it orange? Mm-hmm. Orangish segment where you have a big block of the arm that's preserved. Okay, so that shows despite all this divergence that there are still big blocks of chromosomes shared between human and mouse. Alternative splicing, it's not just splicing out the introns, but you can have multiple exons to generate all this diversity in terms of function. So it's kind of a very compact way of generating a lot of different forms of a protein product. All right, now this one, I have a big orange box around it, and I told you there's going to be a question. So please, if you don't understand this concept, does everybody understand it, or is it kind of fuzzy? Which one? Next slide, regulatory genes and proteins. Nobody wants to hazard a... I think it's like the coolest slide, and then...
Yeah, I don't understand it.
Thank you, Ben. Thank you. You're a brave man. Thank you. Appreciate it. Okay. The whole point of it is, I do that regularly, but I made a fool of myself. I said, do I look like an egg? Right? And I don't, because we already saw that there's not that much difference in the protein coding portions, right? So why don't I look like an egg? Because of these regulatory regions. They've mutated, right? And this is going to change then when and where and how much they're turned on or off. Okay? And so you saw that these elements have multiple inputs even. So it's and you saw the dramatic effects of moving a gene adjacent to one of these, right? You've got the leg coming out where there used to be an antenna. So these regulatory elements are what drives morphological change. Full stop. Not the protein coding differences. Okay? So I don't know if I said this, but I've made Drosophila bone morphogenetic protein. I refolded it when I was in Switzerland. Drosophila doesn't have bones. They use this protein to pattern their limbs. It's called decapentaplegic, 15 appendages. It's for patterning limbs. Later it's used for bone and tooth formation. So you can take the Drosophila protein and you can take like collagen, bind it to collagen and plant it subcutaneously in a rodent, and you get a big knot of bone, heterotopic bone. So, and the rat bone morphogenetic protein 2 and ours, identical. Okay, we don't look like rats, we don't have rat teeth, right? So it's not the protein, it's the regulatory regions, okay? This is a big concept, one of the biggest come out of developmental biology in the last 10 or 20 years. Okay, and then, yeah, this regulatory innovation, that's just what we just talked about. I mean, there were just periods then where there were differences in the mutation of these elements, different subsets of targets. This other paper talked about the elements as well. Then we get to the ENCODE project. First results in 2007. This final slide pointed out that more than 3/4 of the genome turns out to actually be important for gene regulation. So before, just really thought that it was a much smaller fraction. So a few percent is coding, but more than 3/4 is responsible for regulation, and that was published last month online. Okay.